let's go to more of what this is. Let's take our stretch from natural selection into speciation a little bit. Let's get into that discussion. So I'm going to do that today, and we'll go to the website, and I don't know where I am, AP Bio, here we go. Hopefully, can't teach without the internet. Okay, this is week what? 7. We need week 8. All right, here we go. All right. It'll be all white. Come on, load up. That didn't help. Okay, let's try Reese. Okay, here we go. All right, I think it's in Microevolution, so let's continue. Now, I'm not going to spend any time on new. You should know that new alleles are always from a mutation. Okay, we've been through this. We'll talk about these types of mutations. Mutation rates are low in animals and plants. There's about 1 mutation in 1,000 genes per generation. That's just low. That's why anytime you guys answer, hey, it evolved because, hey, there's a new selective pressure and there's a mutation. Wrong, 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 okay? Selective pressure is just picking an allele that already existed due to mutation that already happened, okay? Saying mutation is your answer for anything evolution way is usually wrong, okay? All right, so we'll talk about these things, okay? But big thing here, as I talked about at home during your NORTs, sexual reproduction can shuffle existing alleles into new combinations. That's what creates diversity. In organisms that reproduce sexually, recombination of alleles is more important than mutation in producing the genetic differences that make adaptation possible. That's huge. Sexual reproduction is a driving force for evolution because of the crossover that occurs. Again, talking about what? Chromosomes in meiosis that shuffle the alleles, okay, and cause you to have some in low combinations even though they're not selected for. Can't say enough how important sexual reproduction occurs. Okay, we need to talk about some basic definitions. A population is a localized group of individuals capable of what? Interbreeding. Therefore, they're the same species. Being a same species means they're capable of having sexual reproduction with themselves. Gene pool consists of all the alleles. Anytime you hear allele, we know the word it means what? It means expressed gene. But I like to think of allele as a gene on a chromosome. I always think back to chromosome. A locus is a fixed in all individuals in a population, and of course a locus just means on a certain chromosome, there's the place for that gene that produces that protein that makes that allele. Again, these are all important definitions. Okay, the frequency of an allele can be calculated. And we've done this already. I'm not going to walk you through this, but understand the frequency is how much. In sexual reproduction, we produce in low frequencies all the alleles, okay, that are possible. They stay in low because they're not selected for yet, okay? And so we've been through this already. Okay, I'm not going to go through the Hardy-Weinberg. I think we're pretty good with that. We're going to make some simple mistakes, but we should know that the Hardy-Weinberg is only works, again, no mutations. That's something we can consider because they happen so slow. The mating has to be random. We cannot start selecting for in our mating, hey, don't like tall guys. Tall guys, you don't get to mate, okay? And therefore, you start eliminating what? Some of my alleles that could be the same as the rest of the population. You can start eliminating alleles in the population. And that's the reason why you have a male lion that looks so much different than a female lion. Why? Because of the evolution that occurred by selectively what? Picking the male with the bigger mane. And why the bigger mane? The bigger hair mane makes the what? Male lion looks like he's what? Stronger and bigger. And why is that important for the female lion? Well, because maybe that lion can do what? Be a better hunter, be a better provider of food. These are not things that just happen by chance, okay? No natural selection. There's no selective pressure. Okay? Extremely large population and no gene flow. Okay? The population size is the chance events we're going to talk about called genetic drift. And gene flow is that, hey, you start having organisms go in and out of your population that can mess up your allele frequencies. So this needs to be understood in terms of Hardy-Weinberg. Okay, and I think we're in a good shape with that based on all the forms I've given you. Microevolution is what the Hardy-Weinberg does that. Now this is new. Microevolution is a change of allele frequencies in a population over generations. That means there's going to be a change in the P or Q of a single gene that we're looking at that's dominant recessive. We know things do stay pretty consistent, but we do know in epidemiology, looking at the population of statistics, that there are some diseases on the rise and some diseases that are dropping. And a lot of these diseases are due to recessive, autosomal recessive conditions. A lot of them are. So we know that there's microevolution going on. We're not evolving as we used to, but there is microevolution going on based upon the occurrence or susceptibility of diseases. But this is important. There are 3 mechanisms that cause your allele frequency to change. Therefore, there are 3 types of things, or 3 things that can cause evolution. Natural selection. I think we've said enough about natural selection and how that works. Genetic drift and gene flow are the 2 other types, okay, of driving mechanisms that will change the frequency of alleles, that will cause evolution to occur. Now, I want you to think with me. We're talking about speciation. What keeps one species different than another? Isolated reproductivity, correct? They cannot reproduce with each other. And what causes that? The change in frequencies of alleles. Okay? That's a driving change, especially if these alleles are tied to your gonads, your sexual reproductive organs, or some behaviors, plumage, that can affect that. So I want you to talk about that. These are the 3 driving mechanisms that will change frequencies of alleles. And you could change the alleles so much that you actually have the death of an allele. You could possibly lose the recessive or dominant chromosome in your population due to these 3 mechanisms. And what if that dominant big A gene, for whatever reason, was important in this reproductive compatibility, right? Clearly we know that dogs can't mate with fish. They're not reproductive compatible. They have what? Moved away from each other so much that their reproductive organs don't match. Well, I'm telling you that these 3 driving forces could force you to lose this dominant gene and therefore not be able to make the protein that produces whatever you may need to support that sexual reproductive organ and or behavior. That's important. That's important. Okay, so keep genetic drift, natural selection, gene flow. These are our evolution mechanisms. However, only one is adaptive. This is adaptive to selective pressure. These happen, okay, based upon founder effect, size of population, and chance. Okay, but they still can do the same thing. All right, so in any case, let's continue on. Okay, natural selection, we've been through this, but it's a differential success in reproduction that results, success, reproduction. What's another word for that? Catchphrase in natural selection.
Fitness?
Fitness, right, your fitness increases, okay, and that success in fitness increases to the next population due to certain alleles that are passed that are what? More beneficial to the current selective pressure. We've been through that, okay? For instance, an allele that confers resistance to DDT increased in frequency after DDT was widely used in agriculture. You're right. You put this chemical onto organisms that have many, many generations, okay? They're able to have a lot more mutations in a shorter time period when they have a lot of generations going on. Remember, one bacteria basically can breed into 1 million in a 24-hour period. Think about that. That's how many generations is that? An incredible number, which means the amount of mutations that occur can. Before we get here, what is the #1 cause of mutations? Anyone know? What's the #1 cause of mutations? That provide the opportunity to make a new allele. Yes?
Reproduction.
What's that?
Reproduction.
I won't say reproduction. You're not quite there. Growth cycle. How many times am I drawing this? I'm going to draw it all year long. This is mitosis right here, right? We have G1, we have S1, and G2. These are checkpoints, right? Once you're past G1, you're getting ready for meiosis or mitosis. And I want to do meiosis, right? Meiosis is we're going to do make what? Haploid cells, right? This is all about meiosis. Duplication of chromosomes happen there, correct? And when you duplicate chromosomes, what you do is take your DNA that's double-stranded and you open it up. And then you add nucleotides to the other side to make two what? New strands. And this mating over time causes a mismatch. Once in a long while, there's mismatches. Now, we have mechanisms in our body that can correct for that. But once in a blue moon, one of these mismatches get through without us fixing it. We have so many ways to fix that, because if that happens too often, we would die. But so we have this thing called proofreading. So when we duplicate our chromosomes, therefore double our DNA, okay, we proofread for the mistakes when we match a certain, like A, you're going to learn, has to go with a T. Okay, and a G has to go with a C. And we'll learn about that. You may have learned about that in some other course in your earlier science classes. But we'll talk about this nucleotide variability that you talked about today. But sometimes there's a mismatch. Instead of a G going with a C, there's a G that goes with a T, and we would fix that normally. But every once in a very long while, that gets through our proofreading process, and that's how mutation occurs. And that could potentially create a new allele. Okay, any case, let's continue on. So this is the first one I want to talk about. We talked about natural selection is an adaptive type of evolution where the frequency of alleles change based upon a selective pressure. I think we've been down that road. The rock pocket mice was an example. The peppered moth was an example. There's so many examples that you should be aware of. All right. Oh, another one is the stickleback fish. Okay, those are the classic examples. What is genetic drift? Genetic drift is another evolutionary mechanism where allele frequencies can change, and they can change so much that you can have the death of an allele. You can either lose that chromosome in your population that supports a reproductive behavior or physical part that can lead to what? Reproductive what? Isolation, that these guys can't reproduce. And there's many ways, and we'll get to that. But in any case, this is about size of the population. Hardy-Weinberg states that you're in a equilibrium process when you've got a what? Large population size. And the reason for that is, hey, there's no chance events that can wipe out all of your A's. For instance, here's a great example that I'm talking about. Let's say your population is ridiculously small, and I have 4 individuals. Here's A, and of course I'm just looking at microevolution, okay? A, here's a heterozygote, and another, and we have a what? We'll do a AA, and then another heterozygote. So let's just say there's 4 individuals. I know an individual needs more than 2 chromosomes, but I'm just showing you the pair of alleles. What if, by chance, because it's a small population, this is possible, that these individuals get eaten by a predator? Okay? And so what did I do to the amount of A in your population? I decreased it significantly, didn't I? All right? Now, or I can think of it this way. Probably a better way to do this. I think I blew it. Let me do it a different way. Sorry. Okay, to make my point across. That was horrible. I feel bad now. I'm over it. So we got. Let's make it this way. We have big A, big A. We have little a, and just by chance, let's say these are the only individuals in our population. 4 people, ridiculously small. But that's how the founder effect happens. A small group move to a new area, and they interbreed with themselves. Now, what happens? This guy gets killed. falls off a cliff, chance events. He was the one that got picked on. What happened in this population to the recessive allele? It disappeared. No longer part of the population. And what if that recessive allele is important? That allele was important or that expression was important. You lose that chromosome. So genetic drift describes how allele frequencies fluctuate unpredictably from one generation to the next. But it has to be a small population. Okay? And I'm going to show you this tomorrow. All right? Genetic drift tends to reduce genetic variation through loss of alleles, just like I showed you here. It's a total random event. Okay? Listen, what are the chances of having 3 girls in a row? It's very possible. What's a chance? But you know that there's a 50% chance of being a boy or a 50% chance of being a girl. In low populations, you can have crazy events where even if I don't lose this person, that's little a and little a, just by chance, okay, I can eliminate that. All right, obviously, it has to be a different scenario. What are the chances? How these guys separate. Okay, now, here's a great example. Make a little example, we can go. Here's generation 1. We have the red, I guess, we have the pink and white. Okay, so we have codominance here. So here's the first generation. 5 plants leave offspring. The ones that get pollinated enough, just by chance, are these guys. Okay? What's the next population? Look at the P. The frequency of the C, the red, was 0.7. That means that the red allele to make red flowers was 70%. And there was 30% of the W allele. In the next population, because only these guys were able to reproduce just by chance, they got pollinated more, okay, they made seeds, or these all could have been pollinated, but let's say the birds ate all these seeds and didn't eat these seeds. Just by chance in low numbers, what happened to the amount of the red allele? It decreased. And then, of course, because you have a lower percentage of red, the next population, there's a chance to even be lower. And what happened? Just by chance, only what? These red flowers got to have their seeds planted or fallen. And all of a sudden, just by random what? Chance in a small population of which seeds which plants get fertilized and which seeds get to the ground and not eaten, we all of a sudden have a swing and Q is 0. What's Q?
The recessive.
The recessive allele is gone. And you'd say the diversity is gone. And this might be more susceptible to death, okay? But more importantly, we're talking about only what? Color of the flower. What if this was about a gene that helps reproductive success or reproducibility? What if this was a gene? What if this allele was what if, certain other flowers? What if it's an animal and the animals were attracted to the light colored and all of a sudden they went dark colored? All of a sudden there would be part of the population that wouldn't want to mate with these guys because they're used to mating with what? A different color animal. Bottom line is it's a loss of an allele, which means that allele, forget plant color, could be due to reproductive success between 2 organisms, and that is part of the reason why speciation can occur in small populations. Not through natural selection, chance events that allow only certain individuals reproduce, which hugely changes the frequency. That's why in Hardy-Weinberg, you have to have huge populations that limit what we call genetic drift. Take care. Have a good day.